Welcome to Data Integrity 103, which is our final module in this data integrity training and certification course. This will be our most complex module, as we will be discussing the concepts of original records, complete data sets, and data accuracy. We find these parts of Alcoa Plus to be regularly cited on FDA Form 483s, often because there is a disconnect between industry's understanding of these expectations and FDA's expectations. In this module, we will attempt to understand these concepts from a regulatory perspective, with the goal of avoiding any misunderstandings that could lead to regulatory concern. First, the concept of original. For an up-to-date definition, we look to the PICS guidance for data integrity and find the following definition. The original record can be described as the first capture of information, whether recorded on paper, static, or electronically, usually dynamic, depending on the complexity of the system. Essentially, the original record is defined by the SOP or process. If the first capture of information is within an electronic system, then a printout cannot be defined as the original record. The reason the concept of original record is so critical is that as soon as it becomes a secondary or tertiary record via a printout, transcription, data transfer between systems, the risk to the integrity of that data is inherently increased. Anytime data flows downstream from A to B to C, in whatever format, the chances of mistakes or errors increase. If you take a moment to think about it, it's just really simple physics. Let's take a look at a recent FDA Form 483 observation where we see this disconnect between the firm's practice for the use of original records and FDA's expectations. Furthermore, you did not retain all original dynamic records obtained during the course of testing on other laboratory equipment. Your viscometer and UV-Vis spectrophotometer had the capability to save data from product/material testing. Despite having this capability, your analysts failed to save the complete dynamic testing data, and therefore the data was not available for review by the FDA investigator. The viscometer is used to measure the viscosity of finished drug products during release testing, and the UV-Vis spectrometer is used to measure ethanol content during raw material testing. In this case, the site most likely printed out the analytical results and defined the printouts as their original record used for data review and decision making, but failed to ensure the electronic record was saved to permanent memory. The site assumed that as long as the printout was available for inspection, the Alcoa Plus principles would be fulfilled. But as we have discussed, this is a false assumption. This is actually two deficiencies cited in one observation. The first is failing to save the original record, while the second is failure to review the original record. In this case, it would be an electronic file and associated audit trails. It is a good case study because it highlights both requirements for original records. Number one, that they are retained. And number two, that they are used for review and decision making whenever possible. There should be very few cases where a secondary or tertiary record, such as a printout, is used for review decision making. Anytime the original record is not used as the primary source for decision making, a risk assessment must be available to justify this departure from Alcoa Plus principles. Again, this can and is done, but would be in limited circumstances, justified using a scientific rationale documented in a risk assessment. Additionally, in our case study, the inspector cites the dynamic nature of the data, which is also mentioned in the PICS definition of original records. A dynamic record means there is interaction between the employee and the record content, such as saving the record to a specific location, integration of a peak, printing the record from an internal memory or database, or pushing data from an instrument like a balance or a pH meter to a LIMS, among many other examples. A static record, on the other hand, is a record that is generally fixed and does not require interaction between the record and the human for use in decision making. GMP examples of static records are traditionally paper documentation, such as the recording of the time of a bioburden sample collection directly into a logbook or worksheet. This is considered static data once recorded. You may think of a pH meter printout as a static record. However, this requires further evaluation to make this conclusion. For example, if the human was required to print out the record from the small internal memory of the pH meter, this involved interaction and would be considered dynamic. There are very few instances in a GMP environment, if any, where electronic records can be considered static. In reality, whether the data is considered static or dynamic is not a black or white decision, and is not something that should be used in a decision tree scenario to determine how to ensure the integrity of data. Because remember, it is the overall process, or what PICS considers the sum total of arrangements, that matter to our patients. In my opinion, it is best to consider all electronic data to be dynamic, until of course the day arrives when all humans are replaced by GMP machines, but I'm afraid that is many years away and probably not feasible in our lifetime. Let's look at another example, where in this case the original record was maintained and available for inspection, but was not used by the firm for review and decision making. The firm does not audit electronic source data and results navigator audit trails of production equipment to ensure all production activities are adequately documented in manufacturing records and to ensure data integrity. For example, on July 17th of 2023, I inspected the run log for these processes and noted a number of runs conducted on October 13th, 2022, prior to the processing of this drug substance, lot so-and-so. The first run was aborted. However, no record of the aborted run was documented in the manufacturing record, and your firm was unable to explain this discrepancy. As these production runs were performed prior to the processing of the drug substance, we might assume for our case study that these were CIP, meaning cleaning, or SIP, meaning sterilization cycles, being reviewed by the inspector. Within the batch record, the inspector found only the second passing cycle documented when compared to the system audit trail. There was no mention of the earlier aborted cycle. Now, the inspector has found a potential for direct patient impact as a result of failing to embrace the Alcoa Plus principles. In this case, making decisions based on information documented in the manufacturing batch record, which is incomplete, rather than making a decision only after reviewing the original record. In the past, especially before publication of regulatory guidance on data integrity, production or QA reviewers often applied their signature of approval on a secondary or tertiary record, such as printouts, from an office sometimes far removed from the manufacturing floor. Typically, signing a paper or electronic batch record without logging into the systems themselves to verify what is documented in the batch record versus the original record. In this case, the original record meaning the data and the metadata such as the audit trail. As we see in our case study, this leads to direct patient risk and should be avoided whenever possible. What if the aborted cycle was due to a serious issue with the equipment? This would require a work order or possibly further investigation by the relevant SMEs prior to use. Was this performed and documented within the quality system? If not, there may be serious concerns regarding the potential contamination of this batch. Could be microbial or chemical. and any batches manufactured after this event using the same equipment, resulting in potential adverse events, product recall, and drug shortage. The PICS Guide for Data Integrity provides an excellent framework for determining a strategy for metadata or audit trail review and is covered in our advanced data governance workshops. Potential issues such as these can be avoided when we embrace the principles of data integrity by understanding how they result in good decision making. This may involve a change in the way data review is performed prior to applying our signature. For example, many companies are implementing source or original record review by both production and QA. And in the case of QA, are creating new roles in their organizations, such as QA on the shop floor, to perform these activities more efficiently and effectively. At this point, we will skip ahead in the order of Alcoa Plus and address now the concept of completeness, before we tackle the final topic for discussion, accuracy. So skipping ahead, let us now examine the requirement for data to be complete to demonstrate data integrity. When making an in-process production decision, releasing a batch of API for human use, reporting a stability result, or whatever GMP activity is taking place, the decision relies heavily on a complete data set. We touched on this in some detail in our Data Integrity 101 module under the theme of scope. For any GMP decision, from the receipt of a raw material to the release of a finished batch, the use of an incomplete data set could lead to serious miscalculation and direct patient harm. A complete data set refers to all data and information collected that is relevant to the decision to be made. This could involve individual data points, audit trail entries, handwritten notes. The list is essentially endless. The expectation is that everything relevant to that decision is considered. There are two categories we will examine to define a complete GMP dataset. Number one is routine data. Number two is non-routine data. Let's start with routine data. Because of the importance of products we manufacture, there must be a risk assessment performed to define what is and what is not relevant to each GMP decision. Considering the vast amount of data that we typically generate during routine production and testing, for example, the extensive audit trails that are generated within a SCADA or a DCS, it would be impossible for production or QA to consider all of this data during a batch release decision, for example. Despite the regulation we discussed earlier, 21 CFR Part 211.192, which requires all data to be reviewed by the quality unit before the batch is distributed. We can purposefully deviate from this regulation if and only if we have a risk-based approach established. Without a risk-based approach, deviation from the requirement of all will not be accepted by the regulator. The firm must first identify the data routinely collected, such as alarms, audit trails, and other data and metadata, and then develop a strategy for review using the principles of quality risk management and patient safety. The PICS Guide for Data Integrity, specifically its focus on data governance, provides an excellent roadmap for success here, drawing on the foundations found in ICH Q9, Quality Risk Management. Let's examine one recent FDA 43 observation where all routine data was not considered prior to the GMP decision. In this case, the decision to release the batch. Specifically, your firm does not have a written procedure describing the selection and documentation of a specific recipe and adjustments allowed to the recipe parameters that are programmed into the tablet counters used for repackaging drug product. The recipe outlines certain parameters, such as these things and these things, that are set and adjusted based on varying tablet capsule sizes, shape, and/or colors. For example, in tablet counter this, your firm has approximately so many recipes entered into the system for various tablet and capsule shapes and sizes. In addition, your firm does not maintain documentation of the parameters that have been programmed for each of these recipes to verify they were within the qualified operating parameters for the equipment. In this case, the inspector found multiple recipes available for selection based on which product is being manufactured. However, there were no written procedures in place to ensure two important factors. Number one, that the recipes are still aligned with the parameters established during process validation studies prior to the start of production. And number two, that any adjustments that are made during production would be documented in the batch record and available for review. As a result of these deficiencies, the decision to release the batch would be made with incomplete data and metadata, resulting in a potential direct patient impact, in this case, a short count bottle. Now, some may consider a short count bottle to be of little importance, but when considering patients with limited access to transportation or those based in rural areas, this issue becomes very serious and should be avoided by using good process design thinking and the principles of data integrity. Let's now take a look at another observation, this time from QC, where failure to establish a strategy for review of complete routine data and metadata was cited. Your data acquisition software and workstations, including the systems used for your chromatography instruments, have not been suitably qualified to ensure that the data generated for release and stability sample testing of drug products, including these things and these things, are reviewed, complete, and attributable. For example, your firm uses validated laboratory software, including the chromatography software to acquire and process release and stability testing data. As part of your firm's review process of the data collected, your procedure Lab 0007 peer review states the peer reviewer will do a 100% check of all data and calculations. And as part of peer review checklist, it further states that the audit trail will be peer reviewed. However, your associate manager explained that the original raw data and audit trails within the chromatography system are not reviewed during the peer review of data. Instead, only a printout of the data is reviewed. The printout is considered a copy of the data, not the original data. The raw data is not electronically signed, locked, or routinely archived to ensure no additional changes can be made to the data. In addition, any conditioning sequences or potentially unreported data are not reviewed to ensure the data generated for a particular project is complete. In this case, only a printout of the final result was reviewed and considered prior to batch release, without evaluation of the context and history provided via the original record, including audit trails. Does the data provided on the printout represent the complete data set required to make a critical GMP decision? The answer in this case is certainly not, as there is significant interaction between the analyst and the dynamic data set taking place within the chromatography software that should surely be reviewed prior to batch release. How many injections were performed? Are there examples of orphan data here? How many times were the chromatograms processed? Was there an issue with the ability to accurately integrate the results due to unstable baselines? The use of printouts in either a QC or production environment when the original records are dynamic and electronic is a serious decision to be made along with a documented risk assessment. Without a risk assessment, following the guidance found in ICH Q9, the firm may have significant blind spots, meaning decisions being made based on incomplete data sets, resulting in direct patient safety concerns. A simple data and process map, along with an associated risk assessment, would quickly identify sources of variability or gaps within this QC process that would need to be added to the routine review procedures as a CAPA. Trial or test sample runs in QC continue to be a significant concern from the regulators around the world and is sometimes referred to as testing into compliance. Without a strategy in place to ensure complete data is reviewed prior to batch or stability report approval, the site will be operating at significant risk of data integrity problems. Now let's review another example where a risk-based strategy for data and metadata review was established but was not followed as expected. SOP 0416, Alarm Response Program, required the alarm acknowledgement within this timeframe, impact assessment within this timeframe of alarm notification, and QA review within this timeframe of impact assessment completion. Your firm failed to respond and follow up on the alarm responses within the timeframe established per SOP 0416. For example, differential pressure alarm for grade C room 1183 on June 24, 2023. The alarm acknowledgement and impact assessment were conducted and entered during the FDA inspection on September 13, 2023. Differential pressure alarm for grade C room 1183 on January 30th, 2023. The alarm impact assessment was completed on February 23rd, 2023. QA review was completed on August 11th, 2023. We see here the importance of data or process ownership, as the prescribed timelines for alarm acknowledgement were not followed. And as a result, routine production continued with incomplete information regarding the current environmental control. If a serious trend existed, but was not identified due to an incomplete data set, the effects could be serious. The PICS Guide for Data Integrity provides instructions on the concept of data ownership, which could have prevented the situation outlined in this observation. Let's take a moment to look at the PICS definition for data ownership. The allocation of responsibilities for control of data to a specific process owner. Companies should implement systems to ensure that responsibilities for those systems and their data are appropriately allocated and responsibilities undertaken. This is an important concept to embrace, as developing risk-based strategies for data and metadata is just the beginning. In the case above, the firm appears to have established a periodic review of alarms, which may be considered non-critical according to PICS and therefore reviewed on a periodic basis, such as on a monthly basis, mainly to identify adverse trends before they become an issue affecting the product directly. Carrying out the review according to the established frequencies, however, requires assignment to a specific person or department, which PICS defines as a data owner. If ownership of a specific task is not assigned, who is to be held accountable for completion? You can imagine how successful periodic review of data sets would be, in this case alarm management, if no one person or group is assigned ownership. Data ownership is much like system ownership, which is a traditional role in GMP production where the responsibility to ensure equipment is calibrated and maintained on time is assigned to a specific individual. Data ownership is very similar, except that their responsibilities are more focused on ensuring Alcoa principles for a given workflow are established and maintained. The second category to be discussed is non-routine data. Sometimes, for various reasons, situations arise during production or product testing that are unexpected. 21 CFR Part 211.192 refers to these situations as unexplained discrepancies. The regulations require that all such instances shall be thoroughly investigated. Typically, this is accomplished via the quality system. For example, an incident, a work order, deviation, or OOS. Firms must establish written procedures to handle such unexplained discrepancies and then ensure that the GMP decisions that follow, for example, closure of the incident, consider all relevant data and/or metadata. It has been the experience of regulators around the world, however, that such details are sometimes documented or recorded outside of the instructions found within the company's written procedures. For example, within personal notebooks, scratch paper, Word or Excel documents, among others. When this is discovered, the regulator is unsure if GMP decisions, sometimes critical decisions, are now based on complete data sets, or are relevant data and metadata not included in the evaluation. The integrity of the GMP decision cannot be determined. The inspector cannot do their job, and the quality of products coming from the facility are questionable, which, as we discussed earlier, is a serious concern from public health perspective. Here is an FDA Form 43 observation where the failure to consider complete data, in this case non-routine data or metadata, during GMP decision-making has led to the potential for direct patient harm. The elite in-house test method for related substances, an analytical test method for these particular capsules, states that the HPLC runtime is this many minutes for this standard. However, for sample set ID 1107, analyzed for impurity degradants on this particular date, the standards were analyzed for this many minutes. The Associated Analyst Laboratory Notebook for these run, QC 700, did not contain any entry or rationale for stopping the run prematurely. Manually ending the run early may have resulted in the failure to include later eluting impurities in the chromatogram. And as a result, this batch could have been released despite contamination and serious adverse events. This is a serious situation, but could have been prevented if the principles of Alcoa Plus were embraced. In this case, ensuring review and evaluation of complete data relevant to the decision being made. Manual abortion of the run would have resulted in an associated audit trail entry and would be certainly relevant to the decision of batch release. To reference regulatory guidance here, this aspect of the audit trail should have been classified as critical according to the guidance found within PIC's Guide for Data Integrity. Critical audit trails must be reviewed before a batch is released for human use. Blind spots like this cannot exist in a GMP environment, as patients rely on us to establish predictive quality systems, rather than reactive quality systems that rely on product recall to prevent harm. Speaking of prevention of patient harm, the last two principles being discussed, completeness and accuracy of our data used for decision-making, are paramount to this effort. When establishing our data integrity and data governance strategies, a focus on these two components are not optional. Of course, the previous Alcoa principles are important, no doubt about that. But I have found that a focus on completeness and accuracy are especially helpful when considering a strategy that will stand up to any regulatory scrutiny, as they naturally or automatically cover the remaining Alcoa Plus principles. The last of the Alcoa principles, minus the plus, is accurate. This is more of a higher level concept and is difficult to demonstrate without a governance approach to data integrity, which, as discussed before, considers the sum total of arrangements that result in data integrity. and is generally demonstrated via a data and process map and associated qualitative risk assessment. Accuracy is determined through process method validation and a risk-based approach, the components of which are outlined in FDA's Guide to Data Integrity as follows. Controls that are appropriately designed to validate a system for its intended use address software, hardware, personnel, and documentation. As you can tell, we are now moving to a more advanced understanding of data integrity, beyond specific and limited expectations such as contemporaneous record keeping or legibility. It is impossible to discuss GMP data integrity in a modern setting, however, without including a data governance approach, as demonstrated here through the expectation for accuracy. The accuracy of our data depends on many factors, each of which must be considered in our data governance strategy. For example, the accuracy of a final assay value, a CQA, depends on many factors, starting with sample collection, through sample preparation and analysis, processing, and ending with the approval of the C of A. Within this dynamic process, there will be multiple instances where hardware, software, personnel, and documentation are involved. It is the responsibility of the firm to evaluate each step along this dynamic process from a patient-centric risk perspective to determine if appropriate controls are in place to ensure the accuracy of the final assay result. As you can see, there is much reliance between accuracy and the previous concepts discussed in this course. For example, data cannot be considered accurate if it was not recorded contemporaneous or is not legible. In the old days, before publication of the regulatory data integrity guidance, it was often assumed that data integrity was ensured via the method validation studies performed according to ICH Q2, titled Validation of Analytical Procedures, Text and Methodology. This guidance, specifically this QC guidance, provides instructions for determination of method linearity, repeatability, and other critical factors. However, this guidance is really only one component within our governance strategy, as it generally does not include a proposed control and monitoring strategy for personnel and documentation. Let's turn now to a 43 example where the sum total of arrangements were not considered by the firm when they established their control strategy for data integrity, resulting in a serious observation questioning the accuracy of data and therefore the quality of product on the market. Manual integration is routinely used for the capillary electrophoresis impurity methods during analysis of this particular product. Integration parameters for the standard test procedure received from the sponsor have not been optimized to reduce the reliance on manual integration. The parameters in the standard test procedure do not result in proper integration, and as a result, the analysts are manually integrating every peak during each analysis of this product. There is no procedure to describe how to correctly perform integration to ensure consistency from analyst to analyst and from day to day. Review of electropherograms showed inconsistencies in how manual integration was applied. For example, during the testing of this batch, the manual integration appeared to attribute less area to these particular peaks when compared to the standard and other samples analyzed on the same day. Is the method qualified? Meaning, has it been evaluated according to the requirements outlined in ICH Q2, which remember is titled Validation of Analytical Procedures, Test and Methodology? Most likely, the answer is yes. However, as mentioned above, this is really only half of the GMP effort required, because ICH Q2, or your compendial method validation guidance, does not generally include a requirement for significant evaluation of personnel and documentation, or the sum total of arrangements. In this example, the inspector has identified significant variability in how personnel are processing the data, with the probable root cause being listed in the 43 as no procedure being available, describing how to actually perform this manual integration and control variability from analyst to analyst or from day to day. As a result, the accuracy of data reported on the C of A, which may be a critical quality attribute, is questionable, as the inspector does not have time to perform a retrospective review of all data for products within expiry. This could take months. With regard to data accuracy, questionable GMP data used for critical decision making is considered a critical observation, meaning serious observation, according to the PICS guidance for classification of GMP deficiencies. In this guide, a critical deficiency is defined as follows: a deficiency which has produced or leads to a significant risk of producing either a product which is harmful to the human or veterinary patient, or a product which could result in a harmful residue in a food-producing animal. In the case above, if this data is considered critical to patient safety, then we could easily see how the regulator may classify this observation as critical and take significant regulatory action. If there is a significant risk, data was historically inconsistent, which appears to be the case as documented in the observation, is there a concern distributed products would be harmful to the human or veterinary patient? If you were the regulator responsible for protecting public health or a potential patient yourself, the decision is pretty clear. The firm has failed to embrace the principles of governance to ensure the accuracy of critical data. It may require heavy regulatory oversight in the coming years to improve their quality system according to modern data integrity expectations. We should also consider an example from GMP production, so that we don't come to the conclusion that data accuracy is a QC expectation only. Specifically, you have not performed a scientifically justified risk assessment defining the critical parameters of the visual inspection processes used in the vial syringe line and the manual 100% visual inspection. The following observations were made. Manual visual inspections. The procedure for the manual visual inspection doesn't include background and contrast information, illumination intensity, inspection rate, or container handling and movement. As we have been discussing, the accuracy of data depends on the sum total of arrangements, namely the hardware, software, personnel, and documentation components included in the end-to-end workflow. The inspector will examine the workflow in detail during the inspection, typically by observing the activities and/or interviewing the SMEs, and then perform a risk evaluation on the spot, which is a skill in which they are highly trained. Once their risk assessment is performed, oftentimes in their head, they will then ask the firm how they are managing these potential risks through either technical or procedural controls. When a risk is not addressed, this often leads to an observation. In this case, several important factors directly related to the accuracy of final visual particulates data were not being recorded, which was identified by the inspector as important to the overall accuracy of the visual inspection results data. And as a result, the inspector could not verify the integrity of data. We have also discussed the need for a documented data and process map along with a risk assessment to justify the process. This is the why behind what we do. This observation highlights the need for such an exercise. A data and process map with qualitative risk assessment would have identified these steps, for example, checking and documenting the illumination intensity as highly important to the overall accuracy of particular counts, and a documentation requirement would have been added to the procedure via a CAPA. Without this evaluation, however, the firm is unsure if their current procedures are meeting the Alcoa Plus expectations, as many of their SOPs may have been written based on someone's opinion or how we used to do things at our other companies. We might call this flying blindly when it comes to regulatory inspection, and can be avoided by embracing the principles of data governance outlined in the PICS Guide for Data Integrity. This now brings us to the end of Data Integrity 103, and also the end of our 3-part course. Congratulations, you're almost finished. In summary, we have covered the rationale behind the Alcoa Plus principles, why they matter to our patients, and why they are also so heavily queried during regulatory inspections. I hope this course has achieved its mission to emphasize the connection between our data and our patients. Please provide any feedback in our final survey. But before we get to the survey, be sure to complete the final examination using the link below. I hope to see all of you in a future Advanced Data Integrity and Governance workshop or webinar, and I wish you all the best on your data integrity and governance journey. See you soon, and goodbye.